Thanks very much for the invitation today. I am sorry Michael couldn't make it today, but yeah, he is poisoning himself somewhere in the world at the moment, seeing if the poison really works. So I will show a short clip. Some of you may have seen the clip before, but it certainly captures what most people feel about exercise, and I think it does it with great emotion and genuine emotion. So that's my two affiliations. I was asked to give a disclosure at the beginning. This is just to tell you where the research funding comes from. So the first line there, you can see that there's a number of public and private company-based research sponsoring the human clinical trials I oversee. I've done some consultancy work on personalised nutrition, and I'm a company director at a small new startup focused on personalised medicine. And I think based on that, you'll gather I'm going to talk about the aspects of personalising healthcare advice today and how far we've come and perhaps how far we have to go, and perhaps more importantly, why we have to do it. Okay, so let's hear Michael's view of what he thinks of exercise and the talk.
Exercise. I know I should, but I don't particularly enjoy it. I begrudge the time, and I never seem to make much progress. So I wanted to find out what I should be doing, how much and why. If, like me, you thought exercise was just about pounding the streets or hitting the gym for hours on end, then prepare to be amazed. I've been meeting scientists who are changing our view of exercise.
Come on, Michael, you can do it!
They've discovered that we all respond to exercise in very different ways.
These guys here, super responders. Keep driving, keep driving.
And I've looked at some extraordinary new research which suggests we can get many of the health benefits of exercise from just 3 minutes a week.
3, 2, 1, and that's you done. There you go.
Perfect.
Woo! It goes against absolutely everything I was taught when I was in medical school and everything I have read since.
And I think that what he captures at the end there is still, sadly, one of the biggest problems is that the current guidelines and the health and guidelines around physical activity and exercise still reflect data and information and opinions, perhaps more importantly, which are 1, 2, 10 years behind the curve. So today, what I'm going to do is cover what I've written down in this statement here. That is, I'm going to cover the fact that our understanding of the link between physical activity and health is evolving rapidly. There is new data, there is new thinking. There's been many surprises, both positive and negative, in the past 5 to 10 years, which again aren't making it through into the public health recommendations. One of the most striking and controversial is that you can actually demonstrate clear adverse responses in individuals. It's no longer possible to say that if you have a patient who comes through the door in your GP's practice, that giving them a particular exercise program will, for example, reduce their blood pressure. You just cannot say that. Of course, I'm then going to conclude that the only way forward is therefore to test that individual for their likely beneficial outcomes and give them the right advice. This is very much in the same line as what the pharmaceutical industry is attempting to do or talking about, which is tailoring the drug choice to the individual so that the individual actually responds to that individual treatment rather than just using group average data. So what's a healthy lifestyle? That's up there. So I'm left-handed, so I'm all over the place. I got that from a vending machine in Sweden 2 weeks ago. It says, sack your personal trainer, drink this, what is sugary fruit juice. Unfortunately, that's the sort of public health images that don't just appear in commercial products, but they're really coming through even into our public health guidelines. So there's one example of a problem. I don't know whether the personal trainer or the frush juice is more dangerous for you, but I guess we'll cover that in the talk. So here's some general points. Don't smoke. Not very contentious, I think. Maintain a good body mass index. The exact figure is not entirely clear from epidemiology. Do regular exercise, and this is really what I'll focus on today. Do 150 minutes or more of aerobic exercise, and you'll get a variety of health benefits. The question is, where does this evidence come from? And perhaps more importantly, if you can't be bothered to exercise much and you can't be bothered to be very strict with your diet, and you begin to get this message that, well, life can be rather tedious if you're trying to control all of these things simultaneously, what you'd really like to know is which of these behaviours will give you the most benefit, and then maybe just focus on that and enjoy the rest of your life. Okay, so where does the evidence come from? Well, this is one of the clearest sort of examples in the epidemiological literature. These are two examples. I could give you probably 100, and they stretch back several decades. So New England Journal of Medicine publications, and what it's demonstrating is that there's a relationship between exercise capacity and exercise behaviour and health. So exercise capacity correlates with health outcomes. People who report doing more exercise have a better health. And what you do with this data is you adjust it for things that you know about, confounding variables. You can't adjust it for things you don't know about. But perhaps one of the most contentious issues are problematic features of these types of articles, and this is across epidemiology, is the word prediction is used. These are simply correlations. And from my personal view, this is the weakest form of evidence-based medicine. And this leads to some obvious statements. No matter how large the study is or how often an association is reproduced, correlative data do not prove that if you take that individual walking through the door in your clinic or your GP's practice and intervene, put them on drug X or make them do lifestyle Y, that you'll actually benefit them. There's several classic correlation graphs. This one was, again, New England Journal of Medicine. This time I think it was a joke. Perhaps it was Lancet. This is fantastic. This is chocolate consumption per capita. And this is the number of Nobel Prizes you get. So if you take someone down here and you start eating more chocolate, then presumably you're going to get more Nobel Prizes. I think what's interesting is Sweden's an outlier there. They get more Nobel Prizes per capita of chocolate consumption. Okay, overview of the details of the talk. I'm going to focus in on the 150 minutes of exercise a week because it's prevalent across every nation that has guidelines on physical activity focus on this value and this data. I'm going to talk about the impact of exercise on risk factors, formats of exercise, and then go on to see what the exercise actually achieves. So what do the experts tell us? This is taken from the NHS website in the UK. I'm not picking on them in particular, but it's very common. It's a very common sort of snapshot. So it says there, for aerobic exercise to be beneficial, it must raise your heartbeat and make you sweat. So we'll deal with that, but the sweat bit's clearly not true. Do a minimum of 150 minutes a week, moderate intensity aerobic exercise, and if you've got time, combine that with strength training to combat frailty and aging. If you go beyond 150 minutes, you'll gain even more health benefits. So the question is this advice based on solid science, and does it actually work? Well, the first thing to share with you is there are no long-term exercise-only intervention studies in humans. And so we don't have the evidence base to be making these sorts of claims, even for the average individual. What we have is a lot of lifestyle intervention studies which look at long-term outcomes. And so from a funding perspective, if I was NHS or NIH, etc., I'd want this study done. This study would cost approximately about $100 million. We've costed it. And funnily enough, no one's not terribly interested in giving us the money to run it. So what do we know? Well, for sure, there's a large number of supervised intervention studies, and these are the sorts of studies that measure biomarkers of health. So, for example, aerobic capacity, insulin sensitivity, blood pressure, good cholesterol, HDL, bone mass, bone muscle strength. So you can see there on the left all of these potential risk factors and the potential benefits on the right for what they're claimed to be able to do for your public health profile. So how do these factors respond to the 150 minutes a week for 12 to 20 weeks? So this is data, various studies. I'm just showing you a couple as examples. But every study that we've looked at has demonstrated that if you look at it on an individual-to-individual basis, there's a huge heterogeneity in response to physical exercise in humans. So if we look at the left-hand graph here, this is change in resting systolic blood pressure in about 750 individuals, pre and post. So you can see there is a number of people there, and I'll go on to talk about that in a little bit more detail in a minute, that have an increase in blood pressure after the 20 weeks of training. Now, we don't know whether that's because someone's shouting at them for 20 weeks to go and do lots of exercise, or whether it's the physiology of responding to exercise. In some ways, it doesn't really matter from a healthcare prevention perspective. Forcing someone to do something might not necessarily get you the health benefits you expect. Insulin sensitivity, exact same issue. Aerobic fitness, I'll show you more data further on in the talk, but you can actually make about 4 or 5% of people less fit in terms of their oxygen transport with supervised endurance training. Less fit. And we've studied that right down to the molecular level, and we have an understanding why. They simply have a poor or adverse response to developed blood vessels, small microvascular blood vessels. Equally, if we're going to combat aging and frailty, bulk up muscle mass, improve strength, resistance training seems to be the most effective way, except 25% of the population, perhaps even more in the elderly, do not respond and increase their muscle tissue mass. So what's the problem? Well, here's the blood pressure data in a little bit more detail. So here we have 5 independent intervention trials. And these are the percentages of people noted to have an increase or an adverse response in their blood pressure. So that's 2 standard deviations away from the mean response. So it's probably an underestimate. But for sure, there's about 35% of people will not get the expected average decrease in blood pressure with exercise training. So to suggest to them, do all this effort to control your blood pressure and therefore control your stroke risk, clearly doesn't make so much sense. So why has this happened, and what's the underlying reason? So I'm not an epidemiologist, and maybe I'm bashing them a little bit here, but this is what I believe at the moment. I think epidemiologists have confused the poor sensitivity of their methods to detect an effect with the real requirements of exercise to change health biomarkers. So can we achieve some of these same but variable responses with 1/50th of the time commitment? So this is stage 1. Let's deal with the fact that most people won't commit 150 minutes a week to carry out exercise, even if it did work perfectly for them. So over the past 9 years, myself and other colleagues have been publishing what seems like, relatively speaking, unbelievable effects of very, very short, high-intensity exercise. So you'll notice there in the middle, we've got it down to 2 times 20 seconds, 3 times a week. That's work by Niels Vollard, with a time commitment of under 3 minutes. The original protocol was just under 15 minutes. The problem with these first 2 protocols is captured on the far right-hand side. You needed a very specialised ergometer to achieve this training protocol, and these cost in excess of £12,000 or 14,000 euros. So what we've been working through is trying to adapt these methods, go back into the lab, run clinical studies and demonstrate that you can get a protocol that can use a bog standard cycle ergometer, and it can be very time efficient. So what we have here, based on an EU study that we've just completed the clinical phase of, is a protocol which is 4 times 1 minute, less than 9 minutes total commitment. You can do it in your suit. You don't sweat. There's no requirement to sweat. And you can do it on a relatively cheap bike. And I'm going to talk a little bit more about this data. So let me just show you the impact of some of the original studies. So if you take sedentary young people and put them on a 12 to 20 week endurance training program, you don't usually see very much shift in their glucose tolerance. If you put them on this very high intensity protocol, you do see an improvement. So it appears more effective than the 150 minutes a week. If you put diabetics on a modified form of this intermittent training, this work was done in Canada, then you can see here that the area under the curve are the glucose excursions. This is a reasonably well-controlled diabetic, but if you put continuous glucose monitoring on them, you can clearly see that they go above 10 millimolar for a significant period each day. And of course, that's reflecting when they eat. But the effective hit is to reduce baseline plasma glucose and excursions. So most recently, we've completed nearly, I think, 200 subjects through a supervised trial where we're trying to develop genomic predictors of how they respond to exercise, but not just from fitness and glucose, but also appetite, body fat, aspects of how they feel. And what I'm going to show you now is just some of the very preliminary data. So the protocol here is you switch on the kettle, you get on the bike, which is ideally in the corner of the office or the call center or the factory or whatever you happen to work. You do your 3 to 4 1-minute bursts. Kettle's boiled, have your cup of coffee. You contrast this with the classic endurance training clinical trials. What you have there is a daily time commitment that approaches 100 minutes. You have to get changed, you cycle, you shower because you have sweated, and then you're typically hungry. And this is one of the contrasts and one of the things that we're investigating at the moment, the difference in appetite hormone responses to very sharp bursts of exercise versus this consumption-driven idea that if you keep doing more and more exercise, of course you're going to eat more, otherwise you'd fade away. It's very simple. So what's the outcome in terms of fitness? So on the left here, we have the recent data from this clinical trial, EU clinical study. It's only 6 weeks intervention, 4 times 1 minute. We're pushing people at a workload that's just above their aerobic capacity because a cheap cycle ergometer can handle that sort of protocol. And now you can see the distribution of improvements. So that's a gain in oxygen transport capacity, VO2 max, and that is a clear reduction. Some of these people were brought in time and time again during the final week of after testing just to demonstrate this really was a reduction. It's a real reduction. Their ventilation is the same as baseline or higher. The workload that they're pushing out in the last few minutes of the test is the same or higher, but their oxygen transport capacity has actually declined. They have a maladaptation to this exercise protocol. And this is the same as has been noted in many other studies. This is the Heritage study. So this is 750 subjects. And again, there you've got the same 3 or 4% of people having a maladaptation in terms of fitness. Clearly, one size does not fit all. So you can get the same average responses with a few minutes of exercise, but we haven't solved the personalization problem. So just to summarise, you can change biomarkers with as little as 3 minutes of exercise a week. HIIT is interesting in the sense it's self-adapting. You don't need to set up things so that you've got a very fixed percentage of a heart rate or anything complicated like that. You simply give your hardest effort for 20 seconds. You take a small rest and you do it again. You don't have to think too much about doing it. And as I mentioned, at least 20% are still non-responders even with this particular protocol. So the bottom line is 150 minutes is based on only one line of evidence. You can shift the biomarkers using a radically different approach to exercise. We're still left with this question, is that if you do this exercise, be it 150 minutes or be it HIIT, what do you expect to gain? And so I'm going to cover in 2 slides the 2 big ones that have been investigated the most. I haven't touched on cancer, I haven't touched on other conditions because the evidence base there is, frankly speaking, even lower than with these 2 endpoints. So on the way here, I took the underground. Diabetes UK fund a lot of diabetes medical research in the UK and do lots of good things, but this poster caught my eye. So it's making people aware of type 2 diabetes, no problem with that. It makes a couple of very interesting statements, though. Once you have diabetes, you have it for life. And then after this, you can text them and get a free pedometer. So those 2 things caught my eye for 2 reasons. One is that just because you've got impaired glucose tolerance or you're actually, by WHO guidelines, type 2 diabetic, in no way means that you can't reverse that clinical assessment. So I find that scare tactics. You get a free pedometer. Our evidence-based that sending someone a pedometer will help control the type 2 diabetes is very, very poor. So I think we have to be very careful with this sort of almost scare tactics. Once you have diabetes, you have it for life. It's simply not true. So clinical trial data, we've got some really nice data showing lifestyle modifications. So that's exercise plus please don't smoke plus look after your diet better, whatever better means. So there's been several major studies. The DPP trial in the US showed that lifestyle seemed to be better than metformin. The Finnish DPS study showed very clearly reduced appearance of diabetes, substantial. And then most recently, the largest of all the studies, 4,500 people, showed that after several years of intervention, a variety of diabetes-related risk factors were substantially reduced. And by the 10 years, they had actually shown a remission in a number of their diabetic population. So you can go into people with pre-existing metabolic disease, do an exercise and lifestyle program and impact on type 2 diabetes. So that's the good news. Clearly, we don't know how to tailor this for the individual. One of the motivations for the Look Ahead trial, however, was that by reducing type 2 diabetes, you would reduce cardiovascular disease, and that's because the 2 cluster, and this is another epidemiological phenomena. There's a risk, there's a shared risk, but we don't know why. It could be lifestyle, environment, it could be genes. So these are the outputs of those same 2 trials, which were very successful at improving metabolic health and reducing your diabetes risk. I think this graph is really striking. This is the relationship between the intervention group and the control group for the key endpoints in the study. So cardiovascular-related mortality and events, stroke, MI, heart disease. There was no effect. And in fact, the funding was stopped because there wasn't even a numerical trend justifying the further 3 years of funding. And this actually agrees with the 2 previous studies, the DPP and the DPS studies. Lifestyle itself appears to, when applied to middle-aged people, not change cardiovascular outcomes. So that's kind of hard message, because usually we're telling people that exercise is going to be good for you because it stops you getting heart disease or stops you getting a stroke. The stroke one's interesting. If you reflect back on the data I showed you, 35% of people do not get that improved blood pressure control with exercise training, and actually a small chunk you seem to make worse. So is it the case in these particular studies you're actually increasing mortality for some individuals, killing them off, saving others by the intervention? And all we're looking at, of course, is the mean response. So again, if you're prescribing lifestyle changes or exercise changes to an individual, you really need to understand what it's going to do to them. So I think the second paragraph there is really what I'm trying to focus on now, which is this a failure of a one-size-fits-all approach to lifestyle modification? Or is it a failure in epidemiological predictions? So for sure, epidemiology suggested do more physical activity, reduce your diabetes risk, with the caveat that it was lifestyle modification, but it failed in the cardiovascular prediction. So how do we go about predicting responses to exercise? And I'll comment in the last sort of 15 minutes also on aging, because Michael wasn't here. I thought I'd give you some extra data. So I'm not going to give a big overview of diagnostics, but basically that orange-shaped wedge there, that is to give you an idea of a diagnostic performance. So over on the left-hand side, that's the top performance, and over on the right-hand side, that's the zero performance. And beneath that, you can see some different technologies that we can use. So, for example, way over on the far right, you can measure the variation in a single gene. I mean, I don't know about outside the UK, but the BBC every week has found a new gene, or reports a new gene found that cures or explains a chronic disease. I mean, this is completely untrue. For common multifactorial common diseases, there will never be a single gene that can give you more than a few percent of the variance. And so as a diagnostic, you can't use it. To inform the patient, probably not very useful either. And so if you go from the right-hand side to the left-hand side, you can see that yes, you can combine lots of genetic information and potentially have an assay that will give you information, valuable information. And then if you go further to the left, what you can do, rather than measure the DNA sequence changes, what you can do is measure the product of the genome, which is the RNA, that intermediate between the DNA and the protein. And RNA is what we focus on in our clinical intervention studies. This is my son, aged 2.5. He's playing with his first gene chip. It was used already, so he wasn't going to waste a few £1,000. These chips allow you to capture, if you like, the activity of the genome in blood or tissue, and tell you what's been produced, actively transcribed from the DNA. And what you can then do with it is you can put the data into a mathematical model and try and pull out a prediction, which in the first instance is a bit like a correlation. And then what you would hope to do is apply it to prospective studies or blind independent studies to verify it as actually a predictive tool. So here's an example of what we're doing in the EU study. So in the EU study, we have HIT, we have 150 minutes of hellish endurance cycling, we have resistance training, and what we're trying to do is build up baseline predictors of health outcomes. So in this particular study here, we take a biopsy and a blood sample at baseline. We then train people, in this case for 6 weeks, fully supervised. In this case, it was 100% compliant. We ran it in Sweden, in Stockholm, where everyone seems to do what they're told. Determined baseline fitness profiles, determined the reproducibility of these profiles, and then used all of these parameters to try and predict the outcome. Now, we've completed about 1,200 subjects through different collaborations now, so we're really building up our database of being able to predict responses to training in humans. So here's an example. On the left-hand side here is just what's called a heat map. In this case, we've got 29 RNA molecules that we measure in the baseline sample. And if we add these 29 signals together, what we're able to do is we're able to correlate, in the first study, the increase in aerobic fitness. So that's building a model. Then what you have to do is take those markers and then apply it to independent data sets. And so far, we've been able to show that we can predict fitness responses in diabetics, in young athletic individuals. So basically, we can predict a relatively wide range of fitness responses. Really important to say here that if you measure the baseline fitness of an individual, it does not at all correlate with the improvement. And we've seen that in many, many different studies as other investigators. So your ability to respond is different from your baseline fitness level. Of course, you can add other things into this technology-driven approach. You can add in age, gender, whatever, and try and improve the model. But ultimately, what you're left with is a predictive tool to make a decision. Will this intervention achieve the result I hope for in the individual? That's just an overview of what we're doing at the moment. So we've got about $20 million of exercise intervention studies completed, and we're currently going through the bioinformatics phase of the project. We've got different centres, different biomarker technologies. So hopefully in the next 2 years we'll have a clearer idea of how much we can actually predict in terms of human intervention studies. This was funded largely by the FP7 programme. So one thing you might ask is, well, why bother predicting? I mean, fair enough. Here's one good reason. HF Action is another large US-based study. Randomized multi-center trial. Best pharmacological therapy in the heart failure patients, class 2 to 4. And then add on top of that physical rehabilitation, expensive, time-consuming, physiotherapy-driven, supervised exercise therapy. And the idea was to see if this could add over and above the relatively cheap pharmacological strategy. And as you can see there on the right-hand side, on average, it made absolutely no benefit to the hard endpoints in that clinical trial. So it was concluded that it's not worth it, and it's very expensive. So one of the investigators, Bill Krause, one of my collaborators, he had the foresight to measure the training response. So after a few months, look and see, did the individual that we trained actually demonstrate an improvement in their fitness? And then stratify the results based on that. So this time we're looking at results from 1,600 people, so it's a subset. And what he's able to demonstrate is that about a third showed a measurable improvement in their aerobic fitness with the training program. And those people that demonstrated that improvement actually got the improvements in the hard endpoints in the cardiovascular trial. So the answer there is that if you had a diagnostic that said in advance who to put on the rehabilitation program over and above drug therapy, you would save lives. Also, perhaps even more importantly, you perhaps could tell who not to give the physiotherapy to, because you're clearly increasing their mortality. So that's one example, and hopefully over the next couple of years we'll be able to give you a larger number of examples. So I'm just going to cover in the last 5 minutes the recent data we have for biological aging. So choices for a long and healthy life, they're very different between, I've lived in Sweden and Glasgow, grew up in Glasgow, and the choices there are very, very different. This was at the office at the Canadian Longitudinal Study for Age in McMaster, and I took a picture with my iPhone. It's a poster from Bristol Myers Squibb and Sanofi, so it's a relatively old poster. And then it gives you a number of tips for 100 ways to live to 100. Stop smoking, exercise regularly, reduce the amount of cholesterol in your diet, Take up golf. Golf is probably the most frustrating sport in the world. I can't think of anything worse to increase your blood pressure. So here's a number of things that all seem common sense, and many of them are actually within the public health guidelines. So you can have this. All seems common sense. So you can have this. You can choose to buy the voodoo doll, Dr. Feelgood. Dr. Feelgood will help you to de-stress and bring harmony into your life. So it's pretty much some of these things. So I'm not really sure what's nonsense and what's common sense. But one thing's for sure, we all seem to age at different rates. In Glasgow, in some areas, the life expectancy is still close to 60 years for a male in some regions of that city. There's very, very big ranges of longevity. So far, there's been a lot of DNA-based analysis, and there's very few variants in the human genome that can explain human aging. And likewise, we can't explain it by lifestyle very easily either. So what we've begun to do is take the same approach we use for fitness and start to use not the DNA sequence, but the product of the genome, the RNA, and see if we can measure the RNA levels, start to build some models that predict successful aging in humans. So one way to start this is compare healthy, old, 65, older, but healthy, drug-free individuals, and compare them with regular, healthy, young individuals, and just try and build a mathematical model. One of the key things here is in these studies is that we've phenotyped the individuals. We've looked at their function, their physical fitness, etc. We haven't just went into some sort of biobank. The world's obsessed with biobanking everything at the moment. We're paying a lot of money for this biobanking. But you know, it can be very much garbage in, garbage out. You need good clinically profiled samples. So we can build a model on the left, and then we can try and apply that model to a longitudinal study and see, well, does this model actually predict health outcomes? And that's what we've done. So a couple of examples of building the model. So we end up with a model here measuring 0.7% of the number of RNA molecules. We end up with a diagnostic that's about 93%, telling the difference between young and old. Not so surprising. Perhaps more surprising is we go into our databases of clinical data sets and show that none of the RNA markers are responding to exercise, for example. That kind of fits with the sort of long-lived elder studies. But what you can see there on the right is basically the independent tests. So the green bar is where we started. Again, started this work in Sweden. And then we take our diagnostic and we validate it across multiple independent data sets. This is what takes a correlation away from being just a correlation and being something that's a predictive tool. Perhaps more convincingly, we then take it into other organs. So the same signature, the same markers that we found in muscle are able to classify with high accuracy various regions of the human brain. We can also do it for skin, kidney, blood, etc., etc. So one of the take-home messages here is that you can take a person whose passport that says they're 60 years of age, and that person biologically could be the equivalent of the average 50-year-old or the average 70-year-old. So there's a big difference between biological age and calendar or passport age. So to cut a long story short, and that was just a few slides from the project, we can diagnose tissue age, biological age. The question is, does it actually give us any forecasting capacity? So there are very few studies in existence. This is another Swedish study where you've got a healthy sample collected at baseline and then 20 years follow-up. There are cancer studies out there, but usually what they've done is sample the tumour. What we're interested in here is just taking a small sample from the individual and being able to forecast the outcome of the next 20 years. So we had 108 subjects in this particular study, followed for 20 years. We ranked the subjects based on how this gene score was changed in their biopsy by the late 60s. Those that were ranked first had failed to activate the program. Those that were ranked 108th had strongly activated the program. And then we related these to renal function, physical status, and mortality. This is the data we have so far. So on the left there, you've got the relationship between gene score and probability of death. If you are very successful at activating this healthy aging program, then you've got a far less chance of being dead over the 20-year period, converse being true. On the right-hand side, you've got the relationship again between that baseline score and renal function over the following 12 years. So just to be clear here, in this occasion, this is opportunistic analysis. We've taken a small, healthy biopsy from people in their late 60s. So we looked at the RNA profile in the muscle, and that RNA profile is then forecasting mortality and renal function over the following decades. So it kind of indicates that this type of approach is going to be possible. One thing we couldn't find a link between was the gene signature at baseline and physical activity or lean mass. So the muscle link wasn't there. If we look at the markers, these 146, and look and see whether exercise regulates them, this is just a cartoon of the various markers, you can basically see that none are either switched on or off by exercise, either resistance or endurance training. So resistance exercise doesn't reverse aging, either in muscle or probably anywhere else. So again, you've got the question of why bother? Why we really focused on mortality, for example. Surely we should be focused on quality of life, and I think many of these things can help us direct us toward quality of life solutions. But one obvious way of using this sort of information is in terms of organ transplant. So the demand for renal transplant far exceeds the number of available donors. And in order to deal with that, we've been increasing the age limits of donors. But as I've mentioned to you previously, a 60-year-old can have a profile that's under 50 or over 70. So how do we pick? And we think we can use that type of diagnostic for this purpose. Expand the donor criteria and potentially increase this pool here so that we can do more renal transplants. That has huge health economic savings and huge impact on quality of life for the individual patient. Okay, there's a common age signature. We can find it in all human organs. A small footnote, it doesn't relate at all to the aging processes that have been described in C. elegans, Drosophila, and small animal studies. This is distinct and unique to the human. Exercise doesn't regulate it. Again, you can't exercise yourself to the point that you live forever. Summary. All types of exercise yield non- and adverse responders. We haven't found a magic solution. HIIT absolutely deals with the time barriers for modifying risk factors, but it doesn't remove non- and adverse responders. You can modify type 2 diabetes risk factors with 3 minutes of high-intensity cycling in a suit in the corner of your office a week. You can use lifestyle modification to modulate type 2 diabetes, but you won't impact, if you're middle-aged, on your cardiovascular disease outcomes. The public health guidelines for exercise don't work. Time are misleading. They're out of date, in some cases decades. Potentially unethical, because you do have adverse responders, and if you're using exercise to treat an individual, you have to be sure of really what you're doing. There's other considerations as well. If we all follow the 150-minute guideline, we'll all eat or consume approximately 17% more food, most of which is flown around the world. That impacts on your carbon footprint and your wallet. So there's a number of economic issues here as well. And the final point would be that personalised medical advice is required to deliver genuine evidence-based healthcare to the individual. And I think our reliance on epidemiological correlations is perhaps too prevalent in evidence-based medicine. And none of that was in any way new. So this is a GP based in Bath in England, late 1700s. And he said this: "It's much more important to know what kind of patient has a disease than to know what kind of disease a patient has." And I think he was right, and I think hopefully we'll have the tools to solve this particular conundrum in the coming years. Thanks for your attention.
Thanks, James. It's a really fascinating presentation. I think it's reassuring that it's not about having an expensive gym membership that keeps you fit and healthy. We've got about 5 or 7 minutes for questions, and we've had quite a few coming through already. But please do keep sending your questions through. And we've got a couple of microphones around the room. So if you want to ask a question in the room, go to one of the microphones and give us a wave from there. I'm going to kick off with a couple of the questions that we've had. It's been really great learning about all the different aspects around exercise. But for those in the room who do clinical practice, if we've got a overweight type 2 diabetic with poorly managed BP, what's the short message that we can give them that's positive to kind of get them more involved in improving their health?
Well, if you want to give them a positive message, then it's not really about science. It's about empathy for the patient, I guess. If we're talking about science, then what we can say is likely that taking up some form of exercise will help control your diabetes risk. Anything beyond that, I think we're overstating the issue. What we can't be sure, of course, is what to actually prescribe that individual. What's the recipe that we give that individual? We cannot be sure. If they have elevated blood pressure, then we need to understand whether they are in that bottom 15% or adverse responders. And of course, a simple diagnostic, which is why I'm interested in diagnostics, would give you that information.
And how far are we as far as genomics are concerned? So do you think this is going to change over time whenever we're getting our own genome sequence? Is that going to tell us anything about how we should be exercising, what impact it's going to have, and what's most important to us as a patient?
Most of the evidence, most of the activity at the moment is on DNA level analysis, and we actually already have the answer there. For many, many traits, you cannot tell enough from a DNA sample that will ever give you the ability to accurately prescribe or do something for an individual. That's why we're interested in looking at the products, the activity of the genome. Other researchers are looking at metabolite profiles, things that capture more of the variance of the individual. Those are things that you can use as a diagnostic. So it's going to take some time because, if you like, the scientific community is working through the DNA phase, the era. They'll soon get bored with it, realize it doesn't work, and move on to other things.
We'll take a question from number 5, microphone.
Thank you for a fascinating talk. Could you actually clearly describe what the short intensity intervention is?
Sure. So there's several. I mean, that's the part of the problem. But if we look at what we've shown is now, if you like, optimized, you can have 2. One is you can have 1 minute of gentle cycling, 20 seconds maximum effort, 90 seconds gentle cycling, 20 seconds maximum effort. Go make that cup of coffee. That needs to be done on a specialized bike, and what we're trying to do is persuade the manufacturer of that bike to think about mass producing something that that could be used in the office setting and in the factory setting. And we're in discussions with a number of companies that might be willing to pilot test this out in the real world. If you're trying to use just a regular gym bike, then it's 4 times 1 minute on, 1 minute off, and it's at a level 10% above your maximum aerobic capacity. So in order to know that, you have to do a fitness test, get that assessed, get the workload from that fitness test, and then use that thereafter. So that needs a little bit of specialist input. But again, through software links to the bike, recording the work done, etc., cloud-based computing, you can actually automate all that for the workplace as well. And so we're talking about, a a few minutes of distraction in the workplace, then I think that all of that's plausible. That's 3 times a week. That's just because we've done 3 times a week in the trial. We're going on to look at twice a week. Perhaps that's sufficient. But what we're talking about here is what the average result is. So for some people, once a week, twice a week. For others, 3-4 times a week. Maybe the optimum.
Great, and we'll take a question from number 4.
Thank you very much. I'm a psychiatrist. I'm just very interested in a couple of things. One is that all your work seems to be done with adults, and what we know from the work about adverse effects on children and the way that genes are expressed, what's the evidence about patterns of behaviour in children that might allow for a change in those non-responders? Are those RNA response patterns fixed, or can you change that over time, or is it something about children and getting them to exercise? Does that make it any different?
So there is some paediatric work looking at, for example, the efficacy of the high-intensity training. But in terms of personalised responses, there's nothing yet. What I can say is that there's 2 components. Let's take fitness as an example. There are 2 components. There's sort of a baseline untrained fitness component, and that has a genetic contribution. And then you have your trainability, your ability to increase your fitness, and that's thought to be linked to a separate set of genes. At the moment, there's no evidence that we know of that allows you to move from being, say, a non-responder to a high responder, either for your baseline capacity or your training responsiveness. So when we do look at children, it's likely to be the case that it's going to be difficult to move fitness, responsiveness anywhere. That doesn't mean that we can, for example, avoid the adverse responders. That, to me, seems more like, based on some molecular data we've got, to be a maladaptation. And it's possible that you can change that. And we're trying out a few ideas, actually, at the moment.
We're nearly out of time, but we'll take one more question from number 5. And I suspect James will be happy to stay around afterwards for a couple of minutes to ask any other questions. But we'll take one final question.
G'day, Isaac Seidel from Australia. James, thank you very much for an eye-opening presentation, recognising the science behind what you've presented. As a GP, I'd say that it gives me some comfort in having advised patients that running for the bus in the morning or vigorously running up the stairs is still a very good thing. I wonder if you're able to comment on the relationships between exercise and things such as neurotransmitters and brain-derived neurotrophic factor that are coming out of the United States in terms of advising patients about exercise who have depression, anxiety, PTSD and so on.
Yeah. I gave cardiovascular disease and diabetes as the 2 examples because it's the most worn pathway. Of course, there is epidemiological support for the impact of exercise on depression, on various cancers, in particular breast cancer. But the majority of that data is not interventional. And so we're back at the beginning again. Without that intervention-based data and really robust biomarkers, we should be pretty careful about saying exercise is a perfect sort of pharmaceutical type approach to treating anything actually in individuals. Other than, I think, making people feel guilty that they're not doing enough. I think we're pretty sure that it achieves that. So yeah, there's clearly some interest in BDNF release from the human brain with exercise, but again, it's not been linked to any real hard, long-term endpoints like depression, for example. So.
Great. Thank you again for all your questions this morning. It's been a really fascinating hour. I'm sure James will be happy to stay around at the front to take any other questions. Can I just ask you to thank him very much for his time this morning?
Thank you. Thanks.